heredity and evolution. So some portion has been deleted from this, right? So what exactly is heredity? That means maybe my eyes look to my mother and my forehead looks to my father. Okay. So I am taking the characteristics from both my parents, and this is the branch of biology in which we study the hereditary. That means the transfer of characters which are taking place from our parents to offsprings. Okay. So exactly, the DNA is a material. What is DNA? You all know it is deoxyribonucleic acid. If anybody asks you what is DNA made up of, yes, Myra, over to you. Have you heard this
Genes.
DNA?
Yeah, I've heard of it. DNA is made up of genes.
DNA is made up of genes, and DNA is present in over
Chromosomes.
chromosomes. And what are chromatid, centromere? Any idea?
I only know about centomere.
Okay. So in the sexual reproduction, we have discussed about DNA copying. Over here, I am discussing in detail what exactly is DNA. Many a times when we ask to students what is DNA, they generally say it is protein. No, it is not a protein. Okay, so it is a chemical molecule which is made up of genes, as you rightly said, which is responsible for passing characteristics, and it is double strands. You can see over here, it is given by Whitson and Crick. Don't remember this name. Okay, you will study in your higher classes, but this was the model which is called double helical structure. Okay, and this is widely accepted that this is the structure of DNA. So it is doubly strands. Now, if I ask you what are the components to which the DNA made up of, so the functional unit is nucleotides. What is the functional unit? Is nucleotide. And each DNA is composed of three things. First, sugar, deoxyribonucleic deoxy मतलब ऑक्सीजन में से निकाल ली, एक ऑक्सीजन निकाल ली, deoxy, right? So these are of 5 carbon compound. It is not sweet because glucose or fructose is sweet, which is having 6 carbon, but it is only, you can see over here, 5 carbon compound. So it is made up of a sugar which is deoxyribose, right? Secondly, it is made up of nitrogenous base. That nitrogenous base can be A, T, G, or C. I will discuss it. So one is nitrogenous base, second is sugar, and the third is phosphate. Phosphate, PO4 2 negative. Okay, so these are the three things out of which DNA made up of. This is not given in your NCERT. I'm giving you this extra. Now, A stands for adenine. This is the structure. T for thymine, G for guanine, and C for cytosine. So this is the structure, but the base remains the nitrogen. Over here, over here, over here, over here. Don't remember this, but you can remember the names. Okay. So DNA is made up of three things. The function unit is nucleotide, and it can be made up of sugar, that is deoxy, 5 carbon compound, nitrogenous base, ATGC, and a phosphate. Now, can you tell me, is there a charge on DNA?
Charge?
Yeah, is there any charge on DNA or it is neutral?
I don't think so. There should be any charge on DNA.
Okay. So DNA is a negatively charged particle. Why? Because you can see over here, it is a phosphate molecule which is having 2 negative charge. Right? So what is the charge on DNA? Negatively charged. What is the charge?
It will be 2 negative or only negatively charged.
Yeah, it is negatively charged. So it must be attracting somewhere or by somebody, something else, right? If you can see over here, these are the strands, thread-like structure called chromatin. Okay, these are the threads. And it is attracted by this histone protein. Is my slide visible? So these all are the threads are of DNA, and it is attaching itself with, it is attractive with histone protein, right? Clear? Now this is a structure of chromosomes. This strand, these are the two strands. This is one strand or one thread. This is another thread. It is called chromatid. What it is called? Chromatid. And they are also called sister chromatids. The chromosome is composed of two chromatids, also called sister chromatids. Right?
Yes, sir.
Secondly, it is attached with a centromere. This is the place where these chromatids are attached. And this hole is called a chromosome. Okay. So this thread-like structure is chromatid, which is made up of DNA and protein. Now, these are the two strands or two threads, which is called sister chromatids, attached by a centromere and hold together, forming a chromosome. Right. Now, you can see over here, there is a DNA and histone protein all over here. Looking, this stands, this whatever I have drawn over here, this is all over the chromatid. Is it visible? DNA and histone. Now, out of this portion of DNA, functional unit or functional segment of DNA is a gene. Okay. So, a part of DNA is a gene. Is it clear?
Yes,
Chromosome, make up of DNA with histone protein. Okay. Where they are present? At chromatid. Where chromatid is attached, centromere. And what is a gene? It is a small functional segment of a DNA, which give rise to a character. It can be anything. It can be a gene of height. It can be a gene of seed color. It can be a gene of flower. It can be gene of anything. Right? Is it clear? Now,
yes, sir.
Now, this is a very, very important question, and this will definitely ask in your exam up to 2 to 3 marks. What are acquired or inherited traits? What are acquired or inherited traits? So, what are acquired traits? Can you guess? You want to guess something? What are acquired traits? What you understand by this term?
Basically, the characteristic traits that are acquired by birth from the parents.
As it is written over here, characters that are acquired afterward. Like I have learned swimming. Okay. I have gained weight or I have built my muscles. So these are acquired characters which I have gained in this birth, after the birth. Okay. And inherited, that means which characteristics I have taken from my parents, or it is passing from generation to generation. Like many of the person you have heard, they are saying he is diabetic or she is diabetic because his parents are like that. Right? There are some diseases which are called congenital diseases, like myopia and all that. So diabetes, hair color, eye color, these are the characteristics which pass from one generation to another. So they are called inherited, and acquired means which the characters which are acquired after birth. Is it clear? Always mention the example whenever they are asking you the trait. So there are two types of trait, two types of characteristics. What do you mean by traits? Traits means characteristics. These characteristics can be acquired. That means I am getting those things in this lifespan, or if I am inherited, that means I am taking it from either from my mother or from my father. Now there is one more term which is called allele. So if you look for the Google, or you will Google it out, it is an alternative form of a gene. It is an alternative form of a gene. Allele is an alternative form of a gene. But what is a allele? Allele means active gene. Like if I'm taking an example, although it is a bad example, because our skin color does not depend on one gene. It is depending on multiple number of genes. So it can be a bad example, but let us consider. For example, my skin color is brown, or it is controlled by gene A. Okay, it is an allele. Are you listening? Are you with me? Now,
Yes.
suppose you are having a fair color and your gene is A2. So for skin color, we are having two alleles. Two genes are responsible for skin color. A1 for brown, and for fair, it is A2. right? Clear? Let us take what John Mendel has taken. He has taken a pea plant and he has taken the characteristics of height. Let us suppose the gene is A. Okay, gene A is responsible in a pea plant for the height. Now it has been there that either the plant is tall or either the plant is dwarf. Okay, so for particular gene we have two alleles which I am denoting with capital letters, capital T or small t. Now why there is such discrimination, that why I am putting it as capital T or why I am putting it as small t? So for capital T means it is dominant, it express, okay, and small t is recessive, it don't express itself. Like in our class, some students are very active. Whenever teacher asks who want to perform the dance, so they already come up, who want to sing. Some students are very eager to jump into, so they are dominating, right? But suppose the dominating students are absent, then teacher can ask, now who want to sing, who want to dance, who want to participate in dance, who want to participate in drawing. So if the dominating students are absent, then there will be a chance that recessive student can participate. Are you getting my point? So in the similar way, for particular gene, we have two alleles for that. One is dominating and other is recessive. If dominating is there, although recessive is present, although there are 60 students in the class, but only the dominating is expressing him. Similarly, in terms of characters, there is one dominating gene and one recessive gene, which I am denoting with capital letter or small letter. It is just a way to represent a gene. Hope you have understood. So if this capital T is present in a particular organism, then the height will be tall. Although the recessive genes or recessive allele is also present, but it will not express itself. Now, gene is always present in pairs, right? Gene is always present in pairs. Are you with me?
Yes, sir.
Okay, so let us suppose both are tall, both pairs are tall. So both pairs are dwarf. So if dominating is absent, it will be recessive, it will express itself. So that plant will be dwarf. And if both alleles are of same, either are dominant or recessive, they are called homozygous. Homo means same, right? But suppose one dominant allele and other recessive allele, we always represent capital T first, then small t. So it is heterozygous. Clear?
Yes, sir.
Now, if I ask you AIDS, it is acquired or inherited? Is it an acquired disease or inherited disease? Because
It is acquired.
If a mother is suffering from AIDS, definitely the child will get AIDS. Because it passes on, that virus is present in the blood. So it is not inherited. Why? Because although it is from birth, but that trait is not, suppose mother is there, but granny is not having the disease. That is not acquired. It is inherited. That is not inherited. Sorry, slip of tongue. It is not inherited. It is congenital. Right? Now let us take some.
Congenital means acquired.
Yeah,
If it
acquired.
is congenital,
Many times students say that AIDS is inherited, because if mother is having, the child is also having it. So it is not inherited. Okay. Now, what is the variation? As we have seen in the sexual reproduction, that in asexual reproduction, variation is not possible. So the difference among the individual of a species or population, if there is a difference between individual to individual in a population or in a species, it is called a variation. And major cause of variation includes mutation. It can be by just simple reading or misread of DNA when it is copying. So, or gene flow or sexual reproduction, only sexual reproduction show mutation. Now, DNA mutation is a sudden or a spontaneous change in a genetic material due to the various environmental factors like UV rays, ultraviolet rays, or by mistake during DNA replication. Okay, is it clear? What is variation? Now, variation can also be there in a gene flow. What is gene flow? Gene flow, also known as gene migration or allele flow, when it is passing from one generation to another, is the transfer of genetic variation from one population to another. Okay, if it is passing from parents to offspring, what it is passing? The alleles are passing, the genes are passing, the DNA are passing, or they are copying the things. Recombination of a process by which pieces of DNA are broken and combining to produce new combination of genes. Suppose the breakup of DNA is there. You can see over here the offspring migrant has genetic HH, so the red color or pink purple color is there when it is combined. Suppose, let us take an example. Yeah, this is the chromosome of a mother and this is a chromosome father. Now when the crossing over takes place, they attach itself over there. Now they break over here. From there it is breaking, and you can see over here the blue portion is taking the yellow one and yellow portion is taking the blue one. Noted? So what happens over there? Some portion of the DNA are broken and then they are recombining. Right? So in this way the variation is possible, and this exchange is called crossing over. What it is called? Crossing over. Right, now let us see what is homozygous chromosomes. Homo means same, zygous means origin. So in all we have 46 number of chromosomes. So 23 chromosomes are from my father's side and 23 chromosomes are from my mother's side. Okay, now when they are combining, the crossing over takes place. This is father, this is mother, and now the crossing, after crossing over, the traits are transferred, and maybe the allele can be dominating or the allele can be recessive, depending upon the dominant will expressed physically. Are you with me?
Yes.
So what is variation? Variation is caused due to genetic drift, that is change in the allele frequently due to the change in an event. Okay. Now, what is a gene information? Now let us see. A gene is a basic unit, basic genetic unit. Now this is a cell. Inside the nucleus we have chromosomes. Now this is a chromosome. And over here we have DNA and histone protein. Now this is a DNA and this active part. Suppose this is a part for gene of height. Okay. So what is a gene? A gene is a basic unit or genetic information for inherited characters, to determine inherited characters. Now, what informations are there on the gene? Now, gene is a basic genetic information, and genes determine what are the characters to be inherited. It can be height, it can be skin color, it can be hair color, it can be eye color, anything. Now, what is a genome? All this information, the collection of genetic information, is together called a genome. So these are the terms which you need to remember. Which you need to remember is genome, gene, genome, chromosome. What is gene? Gene is a basic unit of genetic information. What is genome? It is a collection of genetic information. Chromosome, a storage unit of a gene. And what is DNA? DNA is a nucleic acid that contains the genetic instruction, specifically the biological development of a cellular forms of life. Any doubt? I'm going a little bit deep.
What is the difference between a mutation and a variation?
After mutation, the variation is possible. When the DNA combination changes, it can be by UV rays, it can be by misread of the DNA, it can be any reason. But if it is passing on to other generation, then it is mutation.
तो म्यूटेशन में भी सेम वेरिएशन ही होती है ना, अगर मतलब डीएनए में मिसरीडिंग होगी तो?
अगर म्यूटेशन होगा तो ही वेरिएशन पॉसिबल होगा, नहीं तो वो एग्जैक्ट कॉपी हो गया ना. फादर मदर का कैरेक्टर था, पर उन्होंने कोई मिसरीड कर लिया, जैसे फादर की आईज ब्लू थी, मदर की ग्रीन वो थी ब्लैक, अब जो बच्चा उसकी ब्राउन हो गई, क्योंकि कोई मिसरीड कर दिया डीएनए। ठीक है, तो वेरिएशन है, ऑल दो फादर और मदर का नहीं है। जैसे फादर का ब्लड ग्रुप ए है, मदर का ब्लड ग्रुप बी है, बच्चे का ए भी हो सकता था, ए हो सकता था, बी हो सकता था, उसका ओ आ गया। ठीक है, तो म्यूटेशन मतलब मैं एक एग्जांपल दे रहा हूँ, एग्जैक्टली ऐसा होता नहीं है, थ्रू एक एग्जांपल आपको समझाने की कोशिश की है मैंने। Now these two terms are there, genotypes and phenotypes. Up till now, you understood everything.
एक चीज पूछनी है, एलील और जीन में मुझे अभी भी कंफ्यूजन है, दोनों में डिफरेंस।
अगर आप बुक में देखोगे ना, तो ये लिखा जाता है कि जीन का ही दूसरा एग्जाम्पल है एलिल। ठीक है, पर टेक्निकली जो फंक्शनल यूनिट है ना जीन की, उसे कहते हैं एलिल, जो फंक्शनल करती है। जैसे हाइट, हाइट का एक अलग एलिल है, इट कैन बी कैपिटल टी स्मॉल टी। सीड का कलर, उसका एक अलग एलिल है, हो सकता है ग्रीन और ब्राउन कलर मिल रहा हो। सीड की शेप, उसका एक अलग एलिल है। टेक्निकली होएगा एलिल और जीन एक ही होता है, राइट। एलिल
यस।
मींस फंक्शनल जीन। अब जीनोटाइप का मतलब होता है एट ईच लोकस, लोकस मींस कलेक्शन ऑफ पॉइंट्स एक्सेप्ट सेक्स क्रोमोसोम। तो दो टाइप के क्रोमोसोम होते हैं हमारी बॉडी में। 46 अगर मेरे पे नंबर है, 23 क्रोमोसोम्स हैं, तो उसमें दो टाइप के क्रोमोसोम्स कौन से हैं, कुछ बता सकते हो? एनी आइडिया?
एक तो सोमैटिक होते हैं
नहीं, वो तो
और एक
नंबर ऑफ, वो तो सोमेटिक और सेक्सुअल्स तुम उसके बता रहे हो, हैप्लॉइड और डिप्लॉइड।
तो आप क्या पूछ रहे हैं?
क्रोमोसोम्स के नाम है, नंबर ऑफ क्रोमोसोम्स के बेस पे नहीं बता रहा मैं।
अच्छा।
क्रोमोसोम 23 जो है, उसमें 22 के अलग नाम है और 23rd का अलग नाम है। चलो मैं आपको बताता हूँ। 23rd पेयर जो होता है उसे हम कहते हैं सेक्स क्रोमोसोम। इट इज ओनली रिलेटेड टू द डिटर्मिनेशन ऑफ दैट सेक्स, वेदर इट इज मेल और फीमेल। और जो बाकी 22 है उन्हें हम कहते हैं ऑटोजोम्स, राइट। दे आर कॉल्ड ऑटोजोम्स. So at each locus except the sex chromosome there are two genes. दो जीन्स बताया आपको हर चीज का, जैसे हाइट का दो जीन्स है। ठीक है, इनको मैं एलएल कह रहा हूँ, फंक्शनल यूनिट ऑफ जीन। दीज कॉन्स्टिट्यूट द इंडिविजुअल जीनोटाइप एट द लोकस, यानी उसका जीन कैसा होगा, उसको रिप्रेजेंट कैसे करूंगा। जैसे कैपिटल टी स्मॉल टी, कैपिटल टी मतलब डोमिनेटिंग जीन, स्मॉल टी मतलब रेसेसिव जीन्स। तो मैं उसको कैसे लिख रहा हूँ, लिखने का तरीका, उसको कैसे एक्सप्रेस करूंगा कॉपी पे, वो होता है उसका जीनोटाइप। और वो अगर मैं लिख रहा हूँ कैपिटल टी स्मॉल टी, ठीक है, अब वो प्लांट जो है वो टॉल होगा या डॉफ होगा? वो टॉल होगा, बिकॉज डोमिनेटिंग कैरेक्टर्स इज देयर। तो फिजिकली वो कैसा दिखेगा, वो होता है उसका फिनोटाइप। तो एक्सप्रेशन ऑफ दैट जीनोटाइप इज टर्म्ड एज फिनोटाइप। उस जीन का एक्सप्रेशन या फिजिकली वो कैसे दिखेगा, दैट इज कॉल्ड फिनोटाइप। पी फॉर फिनोटाइप, फी फॉर फिजिकल। फॉर एग्जाम्पल हेयर कलर, वेट, प्रेजेंस और एब्सेंस ऑफ डिजीज, ये सब क्या है? जीनोटाइप मतलब जीन कैसे रिप्रेजेंट होगा और वो फिजिकली कैसा अपने आप को एक्सप्रेस कर रहा है, वो है उसका फिनोटाइप। इज दिस टू क्लियर? दे कैन बी आस्क्ड व्हाट इज द डिफरेंस बिटवीन जीनोटाइप एंड फिनोटाइप. Let us take an example now. These are the two alleles. Okay. You can see XX, XX, XY, XY. These are the genotype. Now, if XY is there, then the sex of that person is male. If XX is there, the sex of that individual is female. तो ये फीमेल क्या है? फिनोटाइप है। और ये जो मैं लिख रहा हूं ये क्या है? जीनोटाइप। ठीक है, लाइक इसका अब इसमें तो दिखा नहीं रखा, कलर नहीं है, तो ब्लैक और व्हाइट कलर है इसके, दिख रहा है आपको? व्हाइट कलर है, दिख रहा है? सपोज ये क्रोमोसोम ले लो, इसमें ई बी और ई बी, ये क्या और एग्जांपल दिखा रहा हूं आपको। ई बी मतलब डोमिनेटिंग है और ई डब्ल्यू मतलब रेसेसिव है। विजिबल है इसमें? थोड़ा राइट।
विजिबल है।
तो इसमें जो था ई बी एंड ई बी, दोनों डोमिनेटिंग है। तो इसका कलर क्या हो जाएगा? ब्लैक. ईबी मतलब ब्लैक. ठीक है, ब्लैक इज डोमिनेटिंग ओवर रेसेसिव. अब है ईबी एंड ईडब्ल्यू, तब भी ये ब्लैक ही होगा, क्योंकि यहाँ पे डोमिनेटिंग जीन है, डोमिनेटिंग एलएल है, राइट. ईडब्ल्यू, और ये इस पे कोई है ही नहीं जीन, तो ये क्या होगा? व्हाइट दिखाएगा अपने आप को, क्योंकि उस दिन डोमिनेटिंग जीन आया नहीं, तो व्हाइट आई. और ईबी ही है केवल, इस पे कोई है ही नहीं जीन, तो ये ऑलरेडी ब्लैक है. तो ये इसका जीनोटाइप यानी लिखा कैसे गया, और फिजिकली वो कैसा दिख रहा है. ठीक है, लाइक पी प्लांट में जो हाइट है वो टॉल तो है डोमिनेटिंग, और डॉफ है रेसेसिव. ये दो इसके एलएल है. सपोज अ प्लांट इज टॉल, तो दिस इज ऑल्सो टॉल एंड दिस इज ऑल्सो टॉल, but this is heterozygous tall because alleles are different. One is dominating, other is recessive, and this is homozygous tall. You understood the difference between these? This is heterozygous because they are different, and this is homozygous, right? If the plant is having this gene or this allele, then it will be DOF. Otherwise, this will also tall and this will also tall. सो रेसेसिव कैरेक्टर अगर है तो अपने आप को एक्सप्रेस कब करेगा? जब होमोजाइगस रेसेसिव हो. समझ लो, थोड़ा डीप है ये. मतलब
Yes, sir.
की जो रेसेसिव कैरेक्टर है वो केवल अपने आप को तभी एक्सप्रेस करता है जब वो होमोजाइगस होता है. जैसे आपकी
So
क्लास
मुझे इस कंसेप्ट में कोई दिक्कत नहीं है.
जैसे डोमिनेटिंग बच्चे नहीं आए तो आप समझ जाओगी. मे बी यू आर अ डोमिनेटिंग, दैट्स व्हाई यू आर एबल टू अंडरस्टैंड. ओके, चलो लेट्स मूव. अब ये कॉम्बिनेशन है कि ये डीएनए जो है वो कैसे कंबाइन करता है. आउट ऑफ सिलेबस है, एनटीएससी वालों को पढ़ाया था मैंने, इसलिए दिखा रहा हूं. ओके, नाउ यू हैव आस्क्ड व्हाट इज एलिल. सो एलिल इज अ variant form of a gene. So it is a type of gene, right? Some genes have variety of different forms which are located in some position or genetic locus or a chromosome. Each pair of allele represent the genotype or a phenotype. This is clear. Now, let us see the dominating characters. Some dominating characters like purple flower is dominating than white flower. Yellow seed is dominating over green seeds. Round seeds are dominating over wrinkled seeds. Green pods are dominating over yellow pods. So we are discussing the pea plant and what are the dominating characters. Inflated, aerial flowers, tall plants. So these are the characteristics which George Mendel has specified. So George Mendel has conducted an experiment in which he has taken a pea plant. Why he has taken pea plant? Because it is easy to grow and easy available, and its lifespan is also very short. Okay, and he conducted the experiment, but the dilemma is after his death. After 34 years of his death, people came to know what he did. So he is called the father of genetics. Okay. Now, there are some terms which will come in his experiment. This is F1 generation, the hybrid offspring of the parent, then F2, then monohybrid cross. What is a monohybrid cross? Monohybrid cross is that cross in which I am taking care of only one characteristics or one trait. For example, if I am considering only the height, only the seed color, only the seed shape, then it is called monohybrid cross. If I am considering at a time two traits like height as well as seed color or the seed shape, then I am considering dihybrid cross. Okay. Now, what is a test cross? If I am saying F1 parent is recessive or dominating, then we have to test cross it. और बैक क्रॉस इज कॉल्ड, अगर मैं आपको टॉल टॉल कह रहा हूँ कि टॉल टॉल होमोजाइगस है. फॉर एग्जांपल दिस इज टॉल टॉल. It is not changing the color. I want you to interact. Don't sit idle.
सर मेरा गला ही खराब है, मैं कितना बोल पाऊंगी.
Suppose this is tall. So suppose this is tall. Now physically it is appearing that it is tall, but how can I know that it is homozygous tall or heterozygous tall? Because both will show a tall appearance, right? तो मैं इसका बैक क्रॉस करवाऊंगा, इसका सेल्फ पॉलिनेशन अगर मैं करवाऊंगा, सेल्फ क्रॉस करवाऊंगा, तो देखो इसको अलग अलग लिख लेते हैं. अगर सपोज ये है और जब मैं इसको क्रॉस करूंगा, इसको इससे क्रॉस किया तो टॉल आया, टॉल. पर अगर एक भी डॉफ आ गया, एक भी प्लांट अगर डॉफ है, इसका मतलब ये हेट्रोजाइगस है. ठीक है, तो जब ये इसका क्रॉस होगा तो ये दोनों अलग अलग हो जाएंगे, सेग्रीगेट हो जाएंगे, फिर ये आपस में क्रॉस करेंगे. तो कितने कॉम्बिनेशन पॉसिबल हैं वो हम देखेंगे. ठीक है, सो इस केस में 3 तो टॉल हैं पर एक डॉफ भी है. तो अगर कोई भी एक डॉफ आ गया तो आप समझ जाना कि ये होमोजाइगस नहीं है, हेट्रोजाइगस टॉल है. राइट?
यस.
अभी फर्स्ट डे है तो थोड़ा नई टर्म्स आ रही हैं, तो कोई दिक्कत नहीं है, आप थोड़ा स्टडी करोगे समझ जाओगे, इतना कोई हार्ड नहीं है. ठीक है, अब मेंडल ने एक लॉ दिया जिसका नाम था लॉ ऑफ डोमिनेंस. राइट? मेंडल्स लॉ इज स्टेटिंग दैट when two alleles of an inherited pair is heterozygous, then the allele is expressed in dominant, whereas the allele is not expressed is recessive. जो dominant होगा वो अपने आप को एक्सप्रेस करेगा, और जो रेसेसिव होगा वो अपने आप को एक्सप्रेस नहीं करेगा. इसको हम कहते हैं लॉ ऑफ डोमिनेंस. ठीक है, इसको समझने की कोशिश करते हैं इसने किया क्या. चलिए, इसने ये सारा का सारा बहुत सारे प्लांट्स पे किया था, मैं आपको एक ही प्लांट पे समझा रहा हूं. ठीक, उसने पेरेंट लिए प्योर टॉल एंड डॉफ. एक प्लांट था जो कि टॉल प्लांट था, एक प्लांट था जो डॉफ प्लांट था. उसने ये कहा ये P1 जनरेशन है, पेरेंट. ठीक है, आपको कैसे पता चला ये होमोजाइगस है? मैंने इसका बैक क्रॉस करके देखा, अगर सारे टॉल आ रहे हैं तो ये होमोजाइगस है. अब जब इसके क्रॉस हुआ तो इसमें जो जीन्स का कॉम्बिनेशन बना वो ये बना, तो ये है मेरी F1 जनरेशन. ये सारे के सारे टॉल थे. ठीक है, यहां मैंने क्या किया, क्रॉस पॉलिनेशन. क्लियर? अब इनका मैंने करा सेल्फ पॉलिनेशन, तो इनके जीन्स अलग अलग करें. ऐसी बनाना जैसे मैं बना रहा हूं, ये फीमेल, ये मेल. इनके जब कंबाइन हुए तो क्या हुआ, टॉल. व्हाट ही हैज फाउंड कि 3 टॉल प्लांट है और एक डॉफ है. ये क्या रेशियो है, 3:1. राइट? इसको इसने कहा F2 जनरेशन. As I am taking only one characteristic, that is I am taking the height, so it is called monohybrid cross. What is the monohybrid ratio? It is 3:1. Right? If anybody asks you what is the monohybrid ratio done by Mendel, it is 3:1 or 1:2:1. How? This is homozygous tall, this is heterozygous tall, this is homozygous recessive. So this is the genotypic ratio. This is the genotypic ratio, and this is the phenotype ratio. What is the difference? Because 3 tall and 1 dwarf is coming, but out of these 3 tall, 1 is homozygous tall and 2 are heterozygous tall. So they can ask you genotypic ratio and the phenotypic ratio of monohybrid cross. Is it clear?